Hello everyone. This series of classes contain the topic related to the cell biology. So I have already done some classes on plasma membrane. If you have missed that, please go and watch in my YouTube channel, that is Journey of Life Science. So this time I'm gonna take cell biology. If you want topics on molecular biology and genetics, you can go for the Unacademy learning app where I done some classes. So you can search about directly as Mahesh Y. So you can get all free live classes, and you can access molecular biology, genetics, the maximum topics I covered in developmental biology also. So let me learn genes, genomes and chromosomes. So this may contain 8 to 10 classes because I'm gonna make short videos, then you can easily understand everything. So when we see the terms genes, genomes, chromosomes, all are same only. They only made up with the nucleotides. So for example, if you take a gene, the gene is nothing but a stretch of nucleotides. The gene contains a stretch of nucleotides. That's it. The genomes means the whole set of genetic material which is present in an organism or in a cell, which is called genome. For example, if you take humans having DNA and RNA. Here we have DNA as a genetic material, RNA as a non-genetic material, because RNA will participate in the transcription and some of the small RNA molecules which regulate the transcription, translation and other processes. Okay, they regulate some metabolism. So here the DNA which is present in a single nucleus, we can call it as a genome. When we see the viruses, not all viruses, the viruses which having RNA as genetic material, so that RNA we can call as genome for that virus. So the genetic material which is present in a single cell, we can call it as a genome. And the chromosomes, we can see directly in all cells chromosomal structure. The cells which undergo cell division will exhibit the chromosomal structure. That means the chromosome structures perfectly visible during that time. But if you see a normal cell which is undergoing normal metabolism and normal functions, you can't see the chromosomal structure. You can see only the, under microscope you can see only the thread like structures. Okay, so here the genes, genomes, chromosome, everything we are talking related to the genetic material only. So we will talk about the detail about the genes, genomes and chromosomes. So we know that nucleotides are the monomers of DNA and RNA. We can say DNA and RNA are the polynucleotide chains. So one thing we have to remember, DNA does not exhibit only the double helical structure. If you see Psi-174 bacteriophage, which contain the DNA in single strand form, RNA also not always not exhibit the single strand form. It can form the secondary structures. The best example is tRNA. So remember, whatever the genes, genomes or chromosomes, their base is the nucleotides. The nucleotides are two types mainly, purines and pyrimidines. Purines are the adenine, guanine, and pyrimidines are the thymine, cytosine and uracil. Okay, let we see the more about the genome sequencing. What is the meaning of sequencing genome? Just if you will take one cell and you will isolate its DNA. Simply I'm saying one cell, but it is impossible to take one cell exactly. So we will take one cell, we will isolate the DNA or genome which contain in that organism, and we will sequence it means we will read the sequence, that adenine, guanine, thymine, cytosine sequence, how it is arranged. The arrangement of the nucleotide sequence is very important for the functions of a cell. everything is regulated by the genetic material, that we will see deeply in future classes. By the beginning of 21st century, that means this century beginning, so most of the researchers sequenced the genomes of hundreds of viruses, because it is very easy to sequence, because it contain only less amount of nucleotides, or less number of nucleotides are present when compared to the humans. Okay, and Saccharomyces cerevisiae, which is a budding yeast, and Saccharomyces pombe, which is the fusion yeast, and Arabidopsis thaliana, the simple plant. Most of the plant related works first they will do in Arabidopsis thaliana, because they know the genome sequence fully, and they know all the genes what are present in the Arabidopsis thaliana. And C. elegans, which is a marine worm, and a mice. Humans, the human genome sequencing nearly took more than 20 years, and they finally finished it. And the whole uh, world participated in the human genome sequencing, because they took some part of chromosome, some done some part of chromosome, some country done. Okay, and the Drosophila melanogaster, the queen of genetics. So these all, we have all these organisms genome sequencing, which are available, sequences which are available in the databases. The best example is NCBI, where we can access all the type of gene sequencing. Not only this, nowadays they done almost maximum type of animals, major animals. If you see phylas, there are 35 phylas in the animal kingdom. So they done at least one animal from the each phyla also. Okay, so genome sequencing, what is the help with the genome sequencing, why they done, what it is useful when we know the sequence. So with the help of genome sequencing, they found that is researchers found that previously unknown protein coding genes and non-coding genes also. So not only coding genes, there are non-coding genes also, which are present in the genomes of many organisms, importantly eukaryotes. If you see humans, 98.5% of human genome is just made up with a non-coding sequence. So why it is formed like this? What is the use with non-coding sequence? So at the earlier times, when they found there is a lot of non-coding sequence in the human genome, they found it's a junk DNA. Okay, so let me discuss that. So this non-coding DNA also contain the repetitious DNA. Repetitious DNA means it's having many repeats. So if you see AGTC, I'm just saying a 4 nucleotide example. This sequence will repeat continuously up to some 10,000 nucleotides. Like that, there are a lot of repetitious DNAs which contain from the 2 nucleotides to the 2000 nucleotides also, each repetitive DNA length. Okay, so these repetitive DNA is similar in all the individuals of the same species, but not identical. That means, for example, if you take humans, we all are Homo sapiens, right? So we have non-coding sequence which is similar in sequence, but not exactly identical. That means if you take two persons who are not even related to each other, if you compare their non-coding sequence, it's quite opposite to each other. That means there is some similarity, but exactly not identical. Okay, so and another thing also, for example, if some of the non-coding sequence, if it is present in the some sixth chromosome in my genome, it may be present in the fifth chromosome in your genome, because our ancestral lineage means not I'm going to the species, our forefathers and their ancestors are different. So that's why that will change. Why it is changing? We all are Homo sapiens. Then why it is changing? Can we form a new species if it change like that? No. the non-code mutations in the non-coding sequence is sometimes advantage, sometimes a disadvantage, but evolutionarily it is very important. Let me see that. Okay, so the early scientists, they considered the non-coding DNA as junk DNA, and they said that no purpose, there is no purpose with that. Only the 1.5% of coding sequence is enough to form the proteins. After that, they found in the non-coding sequence there are regulatory sites. Regulatory sites means if a DNA sequence want to convert into mRNA through transcription or maybe any other type of RNA, it require a regulatory sequences like enhancer, promoter. These all are present in the flanking regions. Flanking regions means the both sides, 5 prime side and 3 prime sides of the genes. So this flanking sequences contain the regulatory sequences. These are very important. We thought one of the time there is no use with that sequence, but if a RNA polymerase want to recognize a gene or if it want to bind to a gene, it should bind only to the promoter region. Okay, so and along with that, this junk DNA shaped the multicellular genomes throughout the evolutionary process. Example, mobile DNA elements. Mobile DNA elements. Within our genome, there are mobile DNA elements that can move from one place to other place. That's why if I compare with any other person who is not related to me, I can see the mobile DNA elements which are, I mean, some of, not mobile DNA elements, some of the sequences which are not present at the same positions in both of us. I mean, I can agree up to the coding sequence, which is useful for a organism development and its normal functions. But I'm saying about the non-coding sequences. This non-coding sequence is okay, but sometimes if a mutation occurred in this non-coding sequence, it can generate a new gene that maybe leads to a new evolution or maybe death of that individual. So here there is an advantage as well as disadvantage if a mutation occurred in the non-coding sequence. Sometimes this mobile DNA elements will sit inside of the coding sequence, or it will damage the regulatory sequences of a gene, then the gene unable to transcribe, so it will not able to produce a mRNA. So if there is no mRNA, no proteins, so there is a lack of some protein that leads to a genetic disorder. Okay, the DNA in a single human cell, which measures about 2 meters in total length, must be contained within nuclei with a diameter of less than 10 micrometer. The nucleus contain, the size of the nucleus is less than 10 micrometers, but the total length of DNA is 2 meters. See how big it is, and that much big DNA molecule or the genome is fitting simply into the 10 less than the 10 micrometer nucleus. Okay, so the compaction ratio we will say is 10 power 5 to 1. So it's very high. So that means it's a very compacted structure. So there are some proteins called histone proteins. These proteins will form the DNA will wrap around these proteins, and it will form the, it will fit into that less size nucleus. Okay, so the DNA and protein complex, that histone protein complex, we will call it as chromatin. And if you want to see the individual chromosomes, means we can go for the mitosis. So that's why we will do the experiments at early stages like UG and 12th. So we will cut the TS of the onion roots, because the edge cell is undergoing the cell division, that is a mitosis. So it can show the chromosomes like this. This is the human chromosome karyotype, because it contains 23 pairs of chromosomes. Okay, let me discuss more. So if you want to learn about genetics, you can go for the Unacademy Learning app and you can search in my classes, that is Mahesh Vai. Okay, see the simple thing, the DNA is folding around the proteins, histone proteins. The single protein, that is histone protein complex, which is wrapped with the DNA, is called nucleosome. Okay, which is called nucleosome. So the entire DNA with entire proteins in a nucleus is called chromatin. Okay, see how much it is folding more, how much folds it is undergoing, and finally it is forming the genome threads. Okay, so molecular structure of the gene. In molecular terms, a gene is commonly defined as the entire nucleotide sequence that is necessary for the synthesis of functional gene product. So one of the time most people said that a gene is nothing but a nucleotide sequence which is required for the synthesis of a protein or a polypeptide chain, but not, they are simply now saying functional gene product, because the genes will not only produce the proteins through mRNA, they can produce the different type of RNA molecules which regulate the transcription, which participate in the transcription, like if you see tRNA and rRNA which participate in the translation, and if you see the some small RNA molecule which regulate the transcription, because that is very important. There are a lot of small RNA molecules which can like microRNAs, siRNAs, SNO RNAs, which can control the cancer cells also. So everything is with a purpose only they made. Okay, so gene means it's nothing but within a chromosome or within a genome some nucleotide sequence which is responsible for the synthesis of, or which is necessary for the synthesis or to produce the functional gene product. So here the functional gene product may be a polypeptide chain which produced through mRNA, or else it is an RNA molecule, that is the final stage of that product is the RNA molecule. It may be tRNA, rRNA or other small RNAs. Okay, so we already discussed that along with coding sequences, the genes also contain some regulatory sequences which are called enhancers, promoters, etc. Okay, if a mutation occurred in this regulatory sequences, that means that the regulatory sequences which control the transcription initiation as well as RNA processing, if a mutation occurred in these regulatory sequences, it affects the normal expression, because if the promoter is silenced, the promoter usually in bacteria it contains Tata box, GC box. If it is mutated, then RNA polymer is unable to bind to the that promoter region and there is no expression of that gene. So everything, every sequence, whether it is coding or non-coding sequence in the regulatory region, is very important to control the transcription. Okay, so if there is a different protein or if there is no protein, a individual will get the distinct phenotypes. Okay, so almost all, most genes are transcribed into mRNAs, which encode proteins. So mRNA will encode proteins, but some of the DNA sequences which also transcribe into RNAs only. So remember, RNAs only, that will not code for proteins. Example, transfer RNA, ribosomal RNA which participate in the translation, microRNA, small internuclear RNA which regulate the mRNA translation and stability, long non-coding RNAs that regulate transcription. These are just an examples. There are a lot of types of RNA molecules. So I have done classes on that specific small RNA molecules also, which is very important for the NET, SET, GATE, any other kind of professional exam in life sciences. See, the learning of subject is a beautiful thing. When we learn something, we will feel amazed. Because see, just as an example, the 2 meters length of DNA, 2 meter length of genome is fitting into less than 10 micrometers. So how endoplasmic reticulum exactly regulating the protein synthesis? After protein synthesis, the nascent polypeptide chain, it does not contain anything except amino acid sequence. It is entering into the endoplasmic reticulum lumen, and that lumen making it into a perfect protein. So when we learn more, we will feel more amazed. So learning subject not for exam. So learn subject and feel yourself the best, because you learn something about some. It's a magic. Cell is a magic. We can't predict how it will turn. For example, if you see a zygote, a single cell fertilized egg simply, that will give you a complete organism. How? Even it is impossible to generate a artificial uterus, because the natural uterus, natural female body knows at which time which hormone it should give, which time which nutrition it will give. So everything is magic only. So when we learn, we feel that beauty. Okay, so learn the subject in that way, instead of getting marks or instead of cracking subjects. When you learn subject deeply, then automatically you will surely get any kind of exam. You can easily crack any kind of exam. So that is one of the important thing we should remember. When we practice 10,000 multiple choice questions also, when we study continuously 10 hours also, there is no use. Study 1 hour, feel that subject, remember, imagine something structures in your mind. In your own way you can imagine. That is a wonderful thing. Then you can remember at any time. You can teach to more persons also. Okay, thank you. I'll continue the classes this week. So within 1 week I'll complete the genomes and chromosomes, and it is very important topic for all the exams. Thank you.